Okay, well, it's probably a little bit overambitious in what I sent in. I'm not going to get to all the things that Bob talked about, but in this, I am going to talk about some things that we're currently doing in the lab, and I hope it's of interest to you. To begin with, I just want to give you a little bit of background. I began my work in this general field a long time ago, working primarily with primates, and I would train them to vocalize, and then we did standard neurophysiological techniques of using microelectrodes to record from neurons in various regions of the brain. And after a while, I began feeling like I wasn't able to really do the types of experiments I wanted to do, and so I wanted to move on to do something that was a little bit more in line with some other techniques. And I went to the other types of motor systems that have been used for years and years. And one of the hallmarks of some of the early work, in fact, going back to the days of Sherrington 100 years ago, was the perturbation technique, namely jerking limbs around and looking at the response to them. It's been done in various avenues, including speech. In fact, some of the early work that I did, we were perturbing the lips and jaw and things like that. But I was still interested in doing the voice. And, but nevertheless, with the perturbation technique, some of the advantages are that you can look at the timing between a stimulus and a response. You can look at automatic responses. You don't necessarily have to rely on volitional types of responses. And you can do some parametric scaling, looking at the gain of the system. And when I say gain of the system, I was toying with the idea of showing all these negative feedback systems in my talk today, but I knew all of you were totally familiar with them, so I'm not going to show you slides of models. Okay? You know what I'm talking about, hopefully. So I was interested in how we could study the voice using this technique. And believe it or not, some of the first work I did is that I took a servo motor, and I was lying down, and I had the servo motor against my larynx. And I was applying pulses, much as a good neurophysiologist would do, to look at some motor system. And it was a dreadful experiment. It just didn't work out at all. And so finally it occurred to me, well, what is the significant thing about the voice? Well, it's an auditory signal. So I thought, well, how do we perturb the auditory signal? And it suddenly dawned on me, well, what we do is we take the auditory signal and we process it and feed it back to the subject in a perturbed fashion. And so that's how I began this work. And all of it falls around the general theme of how is it that we use auditory feedback for voice control. And so what I'm going to do in my talk today is breaking it down into two different components. Behavior, where we talk about just vocalization per se, as well as vocal responses to perturbations in auditory feedback. So that's the basic paradigm. But then lately we've been doing a lot of work on the neural control of these mechanisms, and I'm also going to be talking about EEG recording and analysis that we're doing during these behavioral paradigms, and just a little bit of work on fMRI. We've also gotten into work using transcranial magnetic stimulation. I'm not going to talk about that today. I just thought it was too much. And we're moving in the direction of using integrated EEG and MEG, but there again, I'm not going to be talking about that today. So this slide just briefly shows the experimental paradigm. We have a speaker talking, taking the acoustical output, and we run it through a pitch shifter off the shelf device and feed it back to the subject's ear. And in the lower part of the figure, what we see, this is a trace of fundamental frequency of the voice output. It's not the AC signal. We take the AC signal and we take each glottal cycle, compute the time intervals between pulses and the inverse of that to give us frequency. So this just shows voice frequency as a function of time. It's fundamental frequency. So that's the voice. And then on the lower curve here, this is the feedback. And so the feedback is constant. And at the dotted line, we introduce what we call a pitch shift stimulus. In this case, the stimulus is a downward change in fundamental frequency. The duration of this pitch shift stimulus is about 200 milliseconds. We prefer to use brief pitch shift stimuli like this in order to keep the response in what we would call more of the automatic mode. In one of our first studies, what we did is we manipulated the duration of the stimuli, and we found that if the stimulus was much greater than about 300 milliseconds, we began to get a voluntary type of response. Now, voluntary responses are great. I use them all the time, like now. However, in order to understand some of these basic levels of neural processing, we wanted to keep it at the automatic level. And so we generally use a very brief stimulus. I will say that right now in the lab, we are turning our attention to the voluntary responses because there's a huge amount of work that we've overlooked, and we're turning to that in the future now. But at any rate, with this pitch shift stimulus, we can see that there's an upward change in the voice fundamental frequency, and this is the response that we're interested in. Now, this gives some other examples. First of all, just to orient you a little bit, the black line up here, these are control trials in which a subject was vocalizing when there was no pitch shifted feedback. In the upper case here, we have an upward pitch shift stimulus. The red trace indicates the response to the pitch shift stimulus. And the vertical lines are just the error bars. Now, in the bottom trace, we have a downward pitch shift stimulus. And here we see an upward response. So we call both of these compensating responses, namely because with an upward stimulus, you shift your voice pitch down, and with a downward pitch shift stimulus, you shift your voice up. Okay, so what's going on here is that your motor system is compensating as if it were an error in your production. And this is the way all motor systems appear to work, at least in part. Mine doesn't work too well because, as you can see, this light jiggling around there, that's just reflecting my tremor. A little bit nervous, so it's shaky. And so my nervous system isn't correcting 100% for that, but it doesn't do that for the voice either. So we call this the pitch shift reflex, and it's a way for studying the role of auditory feedback on vocalization. As I indicated, it's a perturbation paradigm. The stimuli are generally unexpected and therefore unpredictable, although we've done a recent study in which we've shown that if they are predictable, then we get differences in the EEG signal, but I'm not presenting that today. The other thing is that this paradigm mimics natural conditions. Namely, you're taking the subject's voice and you're just manipulating it, feeding it back, and you can measure how they react to and correct for errors, and you can quantitatively relate voice output to changes in auditory feedback. Before going forward, I want to go backward to illustrate one other thing, just for sake of clarification. These figures here are very misleading. They're very misleading because what I've drawn here is a very tiny stimulus and a huge response. It's actually the other way around. That stimulus is probably should be way up there or higher, and the response is smaller. But I've just drawn it this way just so you can see the nature of the response. Typically, the responses are about 20 to 30% of the stimulus. They're very rarely at the same magnitude as the stimulus. We have seen that occasionally, but we don't understand the reason for that. I think some people are just hyper-reflexive. So let me go forward again. Now, what I want to do is show you the results from some of our speech studies. A lot of the studies that we've done have been just with sustained vowels. Subjects sit in our sound booth going, ah, over and over again, hearing their voice fed back to them. But then we've had some studies, such as a study we did on Mandarin with Yi Shu some years ago. And we had subjects saying 3 different phrases. In this case, I'm showing the phrase ma, ma. If you speak Mandarin, that's a high tone, 2 high tones in a row. I don't know what it means. But then we also had a high tone followed by a rising tone, or a high tone followed by a falling tone. I'm just showing the 2 high tones. Okay, so there's 2 syllables, ma, ma, and here's the pitch shift stimulus, a downward pitch shift stimulus. And the yellow lines show the control, that is without pitch shift stimulus in both cases. And the blue lines show the response. And you can see that in this case, the subject began responding in less than 100 milliseconds after the onset of the pitch shift stimulus. And with this downward shift, the subject raised his FO as if to compensate. On the other side over here, saying the same phrase, there's an upward shift, and here the subject responds by lowering the FO. Now, one thing it's important to keep in mind is when subjects are doing this, they don't realize that they're making this response at all. If you say a phrase like ma, ma, you are changing your articulators when you go from an ah to an m. Your lips close. The nature of the acoustical signal changes. The fundamental frequency changes a little bit. You can see there's, in the yellow line, there's some changes going on there. So we've got that change going on, but then we've got this time-synced change right there. And so they don't realize they're producing this response, but in fact they are. Okay, now here is an example from a study we did on English speech. We had people saying the phrase, you know, Nina, and we tried to get them to produce it the same way all the time. Before each vocalization, they would hear this standard phrase, you know, Nina, and we tried to make it so that the first part of the phrase was all a steady level FO, and then they just raise their fundamental frequency right at the tail end, na. And what we did then is we introduced these pitch shift stimuli you see at the bottom, indicated by the dashed line. And on the left, there's a downward pitch shift stimulus. The black lines indicate the control trials in which there's no stimulus, and then the blue lines indicate the trials where there's a response to the pitch shift stimulus. So when the subject is preparing to raise the fundamental frequency here, and if the voice auditory feedback is shifted down, they begin early to elevate, simply because what they're hearing at this point in time, starting there, is that their voice is going the wrong way from where they want to go. And so they respond by an early response to increase their fundamental frequency. And on the right-hand side, it's just the opposite. They know this is what they want to say, nah, but at this point, what they're hearing is it's going up too soon, too high, too soon. And so they lower their FO in order not to produce this too early, I presume, and that results in this response. I also want to point out that the same subject, when they're just sustaining an ah sound, the responses are much smaller. Okay, so what this tells us is that not only do people respond to these stimuli during normal speech, but these reflexes are task dependent. That is, they're modulated by the task. And that's one of the things that all of us who are interested in automatic or reflexive types of responses are interested in, is whether or not they can be modulated by task, because that tells us something important about the nervous system. Okay, now we've also begun doing some work in neurological diseases. Here are data from subjects with Parkinson's disease. And what you see here are, well, first of all, on the left, responses to pitch-shifted feedback, but we've also done work by shifting loudness of feedback, which you see on the right. But in both cases, the Parkinson's patients are indicated by red lines and the control subjects by black lines. You can see that the Parkinson's patients produced larger responses than the control subjects, both for pitch-shifted feedback as well as for shifting loudness feedback. Now, I don't know if you're aware of this or not, but one of the symptoms, the speech symptoms of people with Parkinson's disease is that they tend to talk too quietly. A standard phrase you hear one of them say is, "My spouse needs a hearing aid." I'm talking at a normal loudness, but my spouse can't hear me. Okay? They have the impression that they are talking loud enough, but they're not. Also, their voice tends not to have a lot of inflection. It's flat. Okay, so that's their normal speech. Now, what we're showing here is that these subjects, they respond abnormally to these stimuli. Now, we don't precisely know the reason for this. We think, however, that there's an imbalance in the efferents copy of the signal. That is, the output of motor cortex into the sensory system is somehow abnormal, making them think they're talking louder than they are, but we don't understand how that relates yet to the increases in these response magnitudes. But we're beginning a project this summer where we are going to be recording EEG in Parkinson's patients at the same time they're doing this, and we have a way we think we can get at the efferents copy mechanism. Okay, so now I want to talk somewhat about neural mechanisms underlying these responses. And I want to begin by showing you a slide from a study done by John Hood. John was using, in this study, magnetoencephalography, and he triggered the MEG machine off the onset of the subject speaking. And what we see here is that this heavy line represents the response while the person is vocalizing. Now, they recorded the signal at the same time the subjects were producing it, and then they played it back at the same loudness level. And what they saw is that the MEG response was larger during the listening to the signal compared with when they were producing it. Now, John termed this speaker-induced suppression, SIS. And this is fairly common in the motor systems. As you probably know, you can't tickle yourself. Go ahead and try it. It's thought that you can't tickle yourself because your nervous system is suppressing the sensory feedback from your own motor production. Okay, so that's a fairly common thing. It's more widely discussed as motor-induced suppression. Okay, so that's a very important observation that John made. Then that was followed up by a study by Hanks Maldonado and colleagues. And what they did, they record electrical EEG ERPs. And this shows data. The dashed line shows an EEG response to the normal voice. And then when they pitch shifted the voice, they got a larger response. So this is similar to what John showed, namely that normal production is suppressed. But in John's case, it was if you're listening, it's not suppressed. And what Hanks Maldonado showed is that if it's shifted, i.e., if it's not your same voice, then you respond differently. So that prompted us to ask the question, what are the limits of voice deviation for identification of self-voice? And so what we did is my graduate student, Ruzba Baruzman, is that he applied pitch shift stimuli at voice onset of 0, 50, 100, 200, and 400 cents. By now you might be wondering, what's 100 cents? 100 cents is a semitone. It's a logarithmic scale. There's 1,200 cents to an octave. And we use that measure because it gets us away from, well, this subject had a fundamental frequency of 130 hertz, and this person had a fundamental frequency of 220 hertz. How do we compare them? Well, if they're in the sense scale, we can easily compare them. Our stimuli are calibrated in sense, and we look at the response in sense. So he presented these stimuli in this paradigm, subjects vocalizing, and at the onset of voice, the pitch shifter was turned on, so the subject is hearing either his normal voice or a shifted version of that. And this slide shows EEGs, the top row from electrode FZ, frontal midline, the bottom row from central midline, and we have pitch shift to 0 cents, 50, 100, 200, and 500 cents. The dashed line are the responses to passive listening, and the heavy lines, solid lines, are due to speaking. And with 0 cents, you can see that the response to listening is much larger than the response to vocalizing in both these locations. Then as he increased the perturbation magnitude, the response to speaking became larger in this N100 area. Finally, we get out to 400 cents, and we see that the response to vocalization and playback are identical. So this slide illustrates, averaged across all subjects, the relative suppression we see as a function of the pitch shift magnitude at voice onset. And so with zero shifting, there's quite a bit of suppression. But as we increase the magnitude of the shift, that decreases till out to about 400 cents. It's virtually gone. And we now are thinking that when your voice is shifted at voice onset by about 400 cents, you don't recognize it as your own voice. It's like you could tickle yourself when it's not aligned properly. Okay, well, so we've got the suppression at onset. The study we did agrees with the others, namely that self-stimulation results in suppression. And then if it's shifted by about 400 cents, the N100 is almost completely eliminated. And we believe that this is one of the ways in which you identify self-vocalization. In our original models of this system, my colleague Tim Hain understood that when you are speaking or about to speak, one of the first things you have to do is know if that's your voice or somebody else's voice. So one of the first steps in modeling this is to do recognition of self versus non-self. And so we see at voice onset these differences which we believe help to identify self versus non-self. Okay, but then the next question was, is there suppression of responses during vocalization? And so again, here's the experimental paradigm. The subject vocalizes, and then there's a delay time at which the pitch shift occurs. And in this case, the variable, time was randomized, and we had either shifts of 100 cents, 200 cents, or 500 cents interjected. Now, this slide shows the results. It might be a little bit difficult for you to see. The left side is for active vocalization. Passive listening is on the right. And the largest curve is for 500 cents. The dashed line is 200 cents, and the dotted line is 100 cents. Okay, so here we're seeing that when we shift the voice during vocalization, we get something quite different. Namely, the responses become enlarged. But there's something else I want to show you. Here we have, again, 100 cents, 200, and 500 cents. And when we compare the listening, which is the dashed line, with the speaking, we can see that the evoke potential to speaking is much larger than it is to listening. Okay, so this is opposite of what we saw at voice onset. So during vocalization, we see something completely different. Now, to illustrate this differently, Rusba took what's called the normalized difference index, which is the relationship between listening to speaking. And this is really unclear, I'm surprised. This is the P100 response, and, or N100, and this is the P200 response over here. And the black curves illustrate the response to 100 cents shift, and then 200 and 500. Left hemisphere, right hemisphere, left hemisphere, right hemisphere. So what this shows us is that the smaller the shift, the greater the difference between listening and speaking. So it tells us then that, let's just go on to the next slide where you can read this. There's a greater relative enhancement of the signal for small shifts compared to large shifts. In a way that seems intuitive, and it's something that we had suspected all the time. Namely, the responses we get are fairly small. That is, if we have a 100-cent shift, we may respond with a 20-cent response. And so what that means in everyday life is that if I'm going, like that, my voice may be fluctuating by 20, 30 cents, something like that. And I'm responding at about 20, 30 cents. I'm usually not going, which is like a couple notes. Okay, that would be abnormal. So in just normal everyday vocalization, our variability tends to be small. In the same way, when I hold my dot there, it's kind of small. It's not going like that. I'm not that nervous, okay? So our nervous system is geared more towards detecting and correcting for small errors rather than large errors. And as part of this, our voice, our naturally produced voice, we have small changes, as I just said. And if it's a large shift, it's probably an alien voice. And so the system seems better designed for correcting errors in production of your own voice rather than others. Okay, now this shows the topographic distribution of the event-related potentials. And I just want to draw your attention to the difference between 100 cents and 500 cents. Now, even if you're a moderately colorblind person like I am, you can see there's a huge difference here. If you look at these curves, the colors vary quite a bit, which indicates differences between negativity and positivity, and then the actual locations, like this versus this over here. There's quite a bit of difference going on there. So when we go between those two extremes, we're getting much different behavior on the part of the individual, different evoked potentials, and different topographic distributions in the evoked potentials, which tells us that we have some different brain mechanisms going on. Okay, so I've outlined the fact that at vocalization onset, we get the suppression. During vocalization, we get enhancement. There's a timing deal going on. So what's the deal here? So what we did is we did a study where we varied the timing. We presented pitch shifts at start of vocalization, 200 cents delayed, 500 cents delayed, and 1,000 milliseconds delayed. And this shows us some results. 0, 200, 500, and 1,000. Well, at 0, the EEG response to listening and vocalization is about the same, pretty small. But when we go out to 200 milliseconds, the responses are about equal. And out to 1,000 milliseconds, the response to vocalization is much larger than it is to passive listening. And then the same results are graphed here. This shows the P1 response, P100, N100 and P200. And the colors, red and yellow, 0 delay. And so they're, for the most part, about the same. But when we go out to 200, 500, or 1,000, we can see that there's quite a bit of enlargement in these evoked potentials for the longer delays. So there's quite a lot going on between 0 and 200 milliseconds, which then progresses. And we don't know what that is yet. We don't know what the mechanism is. We have a standard negative feedback system model that we use, but we don't have a good timing element in that model yet. Okay, so just to summarize what I've talked about voice onset, where we have identification of the source. After voice onset, we recognize if it's our own voice, and it's sensitive to errors in production and corrects for errors in production. Now, I want to show you a little bit about fMRI. This is the only fMRI slide I'm going to show today. I know you're going to be seeing a lot of that later on. I've been doing this work in conjunction with colleagues in San Antonio, Don Robin, Amy Parkinson, et cetera. And what we did is we did a sparse sampling routine. That is, subjects would vocalize for like 3 or 4 seconds, and they'd receive several pitch shift stimuli while they were vocalizing. And then after they would stop vocalizing, we would turn off the microphone, and then we'd turn on the scanner. And then we would scan for about 3 seconds. And what we show here is we see activations in auditory cortex. And on the looking at the lateral side here, we can see this activation in the posterior part of the auditory cortex in response to this pitch-shifted feedback. Now, I want to show you this slide. This is published by Hickok and Popel. They've been discussing, along with a lot of other people, the sort of a dual stream hypothesis. It comes from the visual system, the what-where differences in pathways. And people have been talking about this in the auditory system as well. And it's been suggested that from auditory cortex in the superior temporal plane up here, there's one pathway that goes towards the ventral part of the temporal lobe and then progresses anteriorly. And this is so-called ventral stream. And just we can think of it loosely as sort of the lexical interface. Then there's a dorsal stream that progresses posteriorly into the junction of the parietal lobe and temporal lobe, and then anteriorly. And it's thought that this is more for sensory motor interactions. And I just wanted to go back to this slide. That's kind of where we're seeing this activation here. So our fMRI activations are supporting this model of Hickok and Popel that this part of the brain is important for interactions between auditory feedback and motor control. Okay, I just have one more study to talk about. And that is, we did a study with musicians. This is done by an undergraduate in our program last year, Nadine Ibrahim. She wanted to look at musicians with perfect pitch, relative pitch, and non-musicians. And so we had our standard paradigm here. And she did testing for perfect pitch, having them read and perform musical scales, et cetera. And there's some standardized tests you do for that. And she recorded EEG signals. And on the left, we have responses to upward stimuli, and the right to downward stimuli. The blue line represents the signal for the musicians with perfect pitch. And then the red line, relative pitch, there. And then the black, the non-musicians. And the thing that really grabs you is this really large 200 response for the musicians with perfect pitch compared to the others. Over here with the downward stimuli for the N100, both the relative pitch and the perfect pitch musicians had a larger potential than the non-musicians. Now, doing the topographic analysis, this is really interesting. Here we have the non-musicians, relative pitch, and perfect pitch. This is in the time range of 200 and 250 milliseconds, and this is a little bit longer time range of 200 to 300 milliseconds. And what we see is that the musicians with perfect pitch, they begin to activate the left hemisphere earlier than the other subjects. Everybody is just doing a lot of the processing in the right hemisphere. But what's unique is that the musicians with perfect pitch begin activating left hemisphere earlier and in a longer time range than the relative pitch musicians begin doing this as well. Now, several years ago, Gottfried Schlaug did some studies using PET and showed that one of the things that's unique about musicians with perfect pitch is that they activate the left hemisphere auditory cortex to a greater degree than musicians with relative pitch. He and nobody else has stated why they do that, but at least these results tell us something. That is, these parts of the brain that are active in the left hemisphere, which would be shown by this N100 here, because that's the negative potential we're seeing here, the blue. That begins earlier in the left hemisphere. And so the people with perfect pitch begin processing corrections to errors in auditory feedback much more in the left hemisphere than do the non-musicians or relative pitch musicians. Now there's another thing. Here are the behavioral responses, the voice responses. Color coding: blue is perfect pitch, red is relative pitch, and untrained musicians. On the left we show, these are grand averages across all subjects. With a downward pitch shift stimulus, the musicians begin an upward response much more rapidly than do the non-musicians. And after the response, the musicians start coming back down much more rapidly than the untrained musicians. It's even more clear with the upward stimulus here, whereas the musicians have a very rapid onset and then they turn it off rapidly, whereas the untrained musicians are very slow. So one of the things that comes with musical training is that when people detect an error, they correct for it very fast, and they turn the response off very fast. I guess it was Heifetz who said, the great violinist, I make a lot of errors, but I correct for them very rapidly. And so this is one of the things that training does for you. I'm sure your daughter's going to correct for her errors as she progresses in her ice skating. Okay, so then just in general to talk about the vocal system. It's similar to other motor systems, utilizes feedback to system control, responses to perturbations. They're small in magnitude, they're short latency, they're directionally sensitive, they're modulated by vocal task. And as far as the neural locations, I haven't shown you much of the fMRI data, but a little bit. We know they're activated in superior temporal gyrus, temporal parietal junction, and also M1 motor cortex. We're also doing work with investigators at the University of Iowa using ECOG, and using the pitch shift technique with them, we're able to show that M1 motor cortex for the laryngeal area is actually activated 30 milliseconds after a pitch shift stimulus. So this is a very tight network we're talking about. I showed you earlier the fMRI work from the sparse sampling showing activation of auditory cortex and the parietal temporal junction. All that is occurring very rapidly, reaching the motor cortex in 30 milliseconds. Obviously, the fMRI results are looking over a very long period of time, and we can't necessarily say that all that's happening within that short time range. fMRI just doesn't allow that. But there is some circuitry that must follow a similar pathway. And I just want to just thank all my collaborators over recent years. The red names are people currently in my lab or collaborators, such as a group in San Antonio, and then a lot of people who worked with me over the years. And no way could I have done this work without the help of all of them, and I'm deeply indebted to them. And thank you. Yeah.
So I was struck by your self-identification study. So 400 cents, four semitones. Consciously, if you listen to your own voice shifted by 4 semitones, I would imagine that you could recognize it consciously. But you're saying that at the N100 level, you don't recognize it as your own voice.
Right. Yeah, I would agree with that 100%. Yeah, you know it's your voice. You can hear it shifted. You're thinking, yeah, it's pretty weird. But yeah, there's mechanisms there, part of the automatic mechanisms that are not recognizing it. I just want to, I think, yeah, we think like you guys do. This is just to prove it. This is our standard model, and things are happening fast here, and we've got efference copies comparing it with auditory feedback, forming a prediction which is then fed back here. There's something going on here about the efference copy. When we have a large disparity right in here, there must be something going on at this point, or at this point where we do some comparison of the magnitude. Some of it automatically, I think, is going on, but then voluntarily there's other things going on. So, yeah, I just don't know what those other mechanisms are yet.
One possibly correlated result that comes to my mind is the result on, I think, on automatic separation of two speakers talking at the same time. I think that becomes essentially a chance when the speakers' pitches are overlapping, are within about a third of an octave of each other. When they're separated by a third of an octave or more, then you start to get, you can start to use FFT-based methods to separate the voices more accurately.
Yeah, there's a lot to that. There's another thing. What I showed you were standard compensatory responses. Some people produce what we call following responses. And I remember in high school, I was on stage doing a performance where I had to sing. And if I was singing solo, it was okay. But if I was singing next to somebody else singing, I would always sing their note. You know, I didn't have my own inborn capability of singing without drifting towards somebody else's pitch. So I follow the pitch of other people. And I don't know what that's about either, but some people do that more so than others.
So that's kind of the opposite direction.
Yep. Yep. Yeah. It's like it's destabilizing. Compensation responses are stabilizing. Following responses are destabilizing. And this is something we don't understand yet. Tori? Or?
You've shown that many groups have advanced responses. Even children have advanced responses. In stutter, they have depressed responses to conditions. Can you speculate a little bit on what might be going on in stutter?
No, I can't. Stuttering is really complex. Language, it involves language, it involves speech. It could just be that they're more sensitive to variations. So it could be part of just the sensitivity of the disparity there. They may have something going on in the transfer to motor commands that produces larger responses. Smaller. Yeah, I just don't know. Yeah, I can't say what it might be yet. There are relatively few groups of us working on this. I know it's an interest of yours, and I hope you can provide us with an answer eventually. But I don't know.
Your fMRI slide, was that a subtraction between the shift condition and the no-pitch condition?
Yeah, we did a subtraction between shift and non-shift, yeah. So this vocalization change between shift and non-shift. Yes?
So you mentioned that during the very beginning, you mentioned that you are measuring the response when the subject is vocalizing, right? And,
yes.
So when the subject is producing the sound, and then you let the subject to hear his own voice through the earphones, actually what the subject is hearing, two voices. One is from the earphone, one is from the bone transduction, right? So, and the signal, the EEG signal you're measuring is actually a signal response from the voice through transduction plus a signal, the response of the voice from the earphones. So how do you eliminate the response to the voice through bone transduction?
It's a real problem. We haven't been able to do it. Nobody else has either. There have been some attempts to modulate filter the voice more in line with what the bone-conducted signal is like. And they've shown the same results. So that's an inherent flaw that we just can't get around easily. That is, the bone-conducted signal has its own separate channel. Now, I will say that if we do a study in which we don't have the auditory feedback very loud, then you can perceptually hear two voices, the shifted and the non-shifted. And that's because the side tone you're hearing is loud enough to get through the earphones. In that case, we don't get much of a response. So that's why when we do these studies, we amplify the signal going to the earphones by 10 dB over what we're producing at the mouth. And then that tends to mask the side tone. It can't completely mask the bone-conducted signal. And so it's an inherent flaw in the paradigm, but nevertheless the results seem to suggest that that is not very important.
Also, I have another question about the fMRI measure. So you mentioned that because, so during the fMRI scanning, we just cannot let the subject produce a voice. So that caused you to, you mentioned that the gateway of the two regions, right, just cannot measure because it just disappears so fast. So have you ever thought about using certain chemicals to disable the person's muscle around the throat, and then let the person to virtually pretend that he's speaking, and then measuring, scanning his brain with the fMRI at the same time?
No, we haven't done that. The closest we came is a study where we anesthetized the vocal folds. That is, we put a tube down through the nose and squirted xylocaine on the vocal folds so that you can't feel them. And when we did that and then did pitch shifting, the signals were larger, which tells us that to make this model complete, this would be for the auditory system. We would have an identical one for the somatosensory system. Okay, and what we think is that the nervous system is looking at two inputs. It's looking at auditory feedback, but it's also looking at somatosensory feedback. And so when we eliminate somatosensory feedback, more weight is placed on auditory feedback. But that's the closest we've come. But we haven't done anything like that in the fMRI. It would be a nice thing to do. The only problem with that is to get good recordings of the types that we've done in fMRI. The subject might be in there for close to an hour, vocalizing repetitively just to get enough signals. And if we spray xylocaine on the vocal folds, it's only going to last a few minutes. So we can't do that experiment now the way we'd like to. A better way might actually be to use PET scanning, where you make several injections onto the vocal folds and do separate scans, but we haven't been able to do that.
Also, do you think that well, I'm just going to propose another solution. Do you think it's possible if we replace the previous method I mentioned with to let a pair of twins with identical voice patterns and let one twin to read a passage to the partner, and then let the partner who is under the fMRI scan present the paper of the passage in front of him, just like the twin under the fMRI is pretending that he's reading.
No, I haven't. That's a novel idea. I guess these identical twins would have to have identical voices too. I think we're going to have an fMRI talk later today. Maybe we can get some light on the possibilities of doing an experiment like that. But it's kind of wild. There's another question back there. Yeah.
Yeah, I was just wondering if you had any thoughts of perhaps the increased left hemisphere activation in the musicians was due to the increased understanding of the syntactic features of music and the increased perhaps, I guess, kind of recruiting the speech and language subject in the hemisphere.
Yeah, that's what's been suggested as far as left hemisphere in musicians with perfect pitch, that it does engage the language components of the left hemisphere. And I think that's a very good suggestion. Our study just looks at the early reflexive issues, and so that, I don't know if it has anything to do with the language components or not. So, yeah. Okay.
Thank you. So now the organizers have scheduled a 2-hour break for a combination of lunch and the poster session. For lunches, to get the lunch that you ordered, you will note that on the back of your name tag is a colored dot, and all you have to do is match up the colored dot with the appropriate colored dots on the sandwiches, on the boxes that are out there. Also, the seating for the lunch is actually in the center part of the atrium, not in the part labeled the Beckman Cafe area. It's in the part with the, basically the symposium logos. The posters are in the east part of the atrium, so please look at the posters from our students. And also remember to sign up for tours of the labs in the afternoon. I suppose if you wish, you can eat your lunches outside, but I would remind you that the heat advisory begins at noon, so eat them relatively quickly. So we'll be back here at 1 o'clock.